Hello everyone, Professor V here, and this is the lecture for Biological Psychology, Chapter 3, Neurophysiology. This chapter is focused on how the neuron functions and communicates with other neurons using the electrochemical signal. Let's get started. Neurophysiology is the study of electrical and chemical processes in neurons. I've been calling the electricity that goes down the axon as the electrical message. However, there is a more proper term, action potential. The action potential is the result of the ion accumulation at the axon hillock, and once the neuron reaches a threshold that is a specific charge, that neuron will fire its own action potential down its axon. More on that in this chapter. We must not forget what neurotransmitters are. They are the chemicals that the action potential is converted into at the axon terminals and enters the synaptic cleft to bind to receptors on the postsynaptic cell membrane. This is a process called synaptic transmission, in which we will also cover in this chapter. Ooh-wee, this is going to be a fun one. This might be a good time to review some of the terminology in which we learned in chapter 2, such as presynaptic neuron, presynaptic membrane, postsynaptic membrane, neurotransmitter, axon, dendrites, soma, and other various terms in which we learned in chapter 2. Okay, let's learn, most likely a reminder of some basic chemistry terms. You may have covered these in a high school or college basic chemistry class. Ions are an atom or molecule that has acquired an electrical charge, positive or negative. There are many floating around neurons in the extracellular fluid, as well as inside the neuron in the cytoplasm. A molecule or atom that has gained an electron is negatively charged are called anions. They are more negatively charged because electrons are negatively charged, and if a molecule gains something that has a negative charge, it becomes more negative. One of the anions that we will become familiar with are chloride ions. A molecule or atom that has lost an electron is positively charged and called cations. They are more positively charged because, again, electrons are negatively charged, and if a molecule loses something that is negative, it becomes positive. There will be a couple of cations that we will become familiar with: potassium and sodium. Now, I mentioned these terms on the previous slide. Ions are dissolved in the cytoplasm or intracellular fluid, a watery solution found within cells. The extracellular fluid is the fluid that is in the spaces between cells. There are ions dissolved in the extracellular fluid as well. The cytoplasm and extracellular fluid is separated by the cell membrane. I like to think of the word membrane as another word for skin in a certain point of view. The cell membrane is like the skin of the cell and is made up of two layers of fatty molecules. So these ions that are in the inside of the cell give the cell a charge. On average, the typical charge of a neuron is between -50 to -80 millivolts when at rest. Thus, this charge is called the resting membrane potential. Now, don't take the term resting too literal because there is a lot still going on. This just means that the neuron is not currently firing an action potential down its axon. As a side note, when compared to the extracellular fluid, the inside of the cell is more negatively charged. So this goes to show that we have real electricity flowing through our brain and body. However, the voltage is measured using millivolts, which is a thousandth of a single volt, because that's how little the charge is, though it is enough for our neurons to communicate with one another. We can actually measure the membrane potential using a microelectrode seen on the next slide. The black electrode would be like a ground wire, and the red is the electrode doing the measuring, referencing the black electrode. There is zero potential when the two electrodes are in the bath, which represents the extracellular fluid. However, when the electrode enters the axon, it records a negative potential. This neuron has a resting membrane potential of -60 millivolts. Again, the inside is more negative than the outside. This is very important to understand when we start talking about thresholds and action potentials in a few minutes. Again, the cell membrane of a neuron is made up of a lipid bilayer, which consists of two layers of fatty molecules. Between the fatty molecules float various specialized proteins, such as receptors that we discussed in the last chapter, as well as ion channels. Ion channels are specialized proteins that open and close to allow ions to pass from the extracellular fluid into the cell and from the cytoplasm to the extracellular fluid. Some remain open and some are always closed, but open at the same time. Again, ion channels allow the flow of ions from inside of the cell to outside of the cell and vice versa. There are many ion channels along the axon in the nodes of Ranvier that we discussed in the previous lecture. Some of them, such as potassium ion channels, stay open, and others, such as sodium ion channels, are gated. Gated means they are closed until the polarity or charge of the cell's membrane at that node reaches the threshold. Of the ion channels that do stay open, most of them are potassium channels. They only allow potassium ions to pass through. This is called selective permeability. This is where the membrane allows some substances to pass through when the channels are open and other substances are stuck on either side. Because the potassium channels remain open, they allow potassium ions to freely pass in and out of the cell. When the neurons has a resting membrane potential, there is a balance of potassium ions on each side of the cell membrane. It is achieved by two forces. Diffusion is the force that causes molecules of substance move from regions of high concentration to regions of low concentration. Think about it like this. When you make Kool-Aid, you pour a packet of flavored sugar in a pitcher of water and the sugar slowly spreads throughout the pitcher. The sugar is highly concentrated at the top at first, but it is spreading because there is a low concentration of sugar in the water. The sugar is diffusion throughout the pitcher. Electrostatic pressure is when diffusion occurs due to ions or molecules being attracted to each other because they have an opposite charge. Think about a magnet. There is a north side and a south side of a magnet. If you place a north side or two south sides of a magnet together, what happens? Yes! They repel each other. Sorry for the Dora the Explorer moment. And if you place the north side and the south side of a magnet together, it attracts. This happens with ions as well. Positive ions attract negative ions and vice versa. However, there are ions that are unable to cross the membrane because there is no ion channel for them and thus attract ions of the opposite charge to them. Let's talk about a specific ion channel, the sodium-potassium pump. This one functions a bit different than the others we talked about. The sodium-potassium pump is always open but always closed at the same time. Okay, saying sodium-potassium pump over and over again is repetitive. I'm just going to say NAK pump from now on. So, the NAK pump is responsible for pushing sodium ions out of the cell and bringing potassium ions into the cell. It does this by being open on the inside. 3 sodium enters the NAK pump, it closes on the inside, opens on the outside of the cell, the 3 sodium ions exit and 2 potassium ions go inside the NAK pump. It closes on the outside, opens on the inside of the cell, the potassium exits the NAK pump, and the whole process happens over and over and over again. Now if that is hard to picture, I understand. Don't worry, I have something that I think will help in a few moments. Neurons rely on electrostatic pressure to bring potassium into the cells as well. There are these negatively charged ions called polyatomic ions inside the cell. There is no channel for them to escape, and because potassium is a cation, the polyatomic ions attract potassium back into the cell to maintain its equilibrium or resting state. You have to remember, the potassium channel is always open as well to understand that this can occur. Here we see 3 diagrams that represent the NAK pump, permeability, and equilibrium, or the resting states of a neuron. You see, as 3 sodium ions leave the cell, 2 potassium ions enter the cell. The NAK pump is constantly working and is requiring a lot of energy to do so. Figure B shows 3 potassium channels on the membrane. Remember, there are other ions dissolved in the extracellular fluid. Because there are no sodium channels on that part of the cell membrane, no sodium can enter the cell. However, there are potassium channels there. This allows potassium to enter and exit the cell because, remember, potassium ion channels remain open. Side notes, the negatively charged gray ions are the polyatomic ions. Notice there are no channels for them either. Figure C shows when there is a balance of sodium on the outside of the cell and potassium on the inside of the cell. The neuron is said to be at rest, and the resting membrane potential for this particular neuron is negative 60 millivolts. Okay, let's move on to the action potential, the electrical message that travels along the axon to the axon terminal. They are very brief. Remember, these messages travel at approximately 220 miles per hour, ridiculously fast. They are large in terms of millivoltage. Remember, a neuron resting membrane potential is in the negatives. During the action potential, the polarity of a neuron shifts well above 0 millivolts. Not only this, but action potentials are sent in a pattern depending on what the message consists of. A certain pattern means one thing, while another may mean something different. Here's a diagram that represents the 2 forces previously discussed. Think of the blue molecules as the Kool-Aid being placed in the water. See how it is concentrated to where it is being poured and slowly is diffused throughout the entire tank. This is diffusion. In the second diagram, there is a semi-permeable membrane with channels. Let's call them Kool-Aid channels. Notice how only the Kool-Aid molecules can pass through the Kool-Aid channels? The other molecules that are in the water cannot pass through because they don't fit or are not compatible with the channel. And the last one demonstrates electrostatic forces. The red molecules are positively charged and the blue is negatively charged. Notice how the red repel each other while the red and blue one attract to one another. This is due to their charge. 2 of the same charge repel each other and 2 of the opposite charge repel each other. When we discuss the action potential, it is crucial to understand these 2 concepts, hyperpolarization and depolarization. So due to the concentration of ions on the inside of the neuron is more negative than the outside of the neuron. Hyperpolarization occurs when there is an increasing negativity of the membrane potential beyond the resting membrane potential. So, if a neuron's resting membrane potential is negative 65 millivolts, when hyperpolarization occurs, the membrane potential gets further away from 0, such as negative 70 millivolts. Depolarization is the opposite. Depolarization occurs when there is a decreased polarization of the cell membrane, meaning getting closer to 0 from the resting membrane potential and even going beyond 0 to make the inside of the neuron positive. This is crucial to understanding how an action potential occurs when we go through the steps in a few moments. So let's say a neuron's resting membrane potential is negative 60 millivolts. When a neuron receives messages from other neurons, these messages accumulate at the axon hillock. In turn, the axon hillock may depolarize, and once it reaches a certain polarization, in this case, we will say in negative 40 millivolts, an action potential occurs in the neuron. The negative 40 millivolts is the neuron's threshold, where the intensity of the polarization is just enough to trigger an action potential to occur. So, what is happening in an action potential is the membrane potential suddenly and momentarily makes the inside of the membrane positive with respect to the outside of the membrane. There is a sudden rush of positively charged ions that come inside the neuron, and this message is sent down the axon. You can see here both the hyperpolarization and depolarization process taking place. When a neuron receives a message, this could cause a depolarization reaction or hyperpolarizing reaction. The resting membrane potential for this neuron is negative 60 millivolts, and the threshold is negative 40 millivolts. When the neuron receives a message and it causes a hyperpolarizing reaction, it causes the polarity of the neuron to move away from 0. If it causes a depolarization reaction, it moves towards the threshold and 0. When the neuron reaches its threshold, this causes an action potential. As you can see, when an action potential occurs, the polarity of the neuron flips and becomes positive, well above 0 millivolts. This is the electrical message. Hey, I'm over here for this slide. I'm over here. Okay, now for what is happening during the action potential. To get a complete grasp of what is happening, you may want to watch this section of the video lecture a couple of times. First, there is an accumulation of positively charged ions at the axon hillock, which is referred to excitatory postsynaptic potentials, which we will cover in a few slides. When the neuron reaches its threshold, an action potential originates in the axon hillock and is shot down the axon. When the electricity hits the membrane of a neuron that contains sodium ion channels, they open. This allows the sodium ions that is in the extracellular fluid to rush inside. Why does sodium rush inside? Because of diffusion and electrostatic pressure. There is way less sodium on the inside of the neuron. Remember, the NAK pump is always pumping out sodium. Also, the polyatomic ions are negatively charged on the inside of the neuron, and sodium ions are positively charged. This is creating an attraction of sodium ions to the inside of the neuron. Because there is all this sodium rushing in, it is depolarizing the cell. Sodium is, again, a cation, meaning positively charged and making the neuron's polarity increase. Also, because of electrostatic pressure, potassium ions are forced out. Remember, both potassium and sodium ions are cations and thus repel each other. Potassium channels are always open, so they are forced out through their own channels. Once the action potential passes the sodium channels close, it is time for the neuron to repolarize or return to a resting membrane potential. With the sodium channels closed, sodium must find another route to get back to the extracellular fluid. This is where the NAK pump plays an important role. The NAK pump, again, pumps out 3 sodium ions for every 2 potassium ions pumped back in. Potassium, on the other hand, is attracted back inside the cell through the potassium channels because of electrostatic pressure. The polyatomic ions are attracting the potassium ions back inside the neuron. This will eventually bring the neuron back to its resting membrane potential. I know that this is a lot to take in or hard to visualize. So, in order to help visualize it, I have included a link to another video on YouTube in the description below that can help with this. It will also help with the understanding of the NAK pump. Perhaps one of these days, I can make my own version. After the action potential occurs, the axonal membrane where it takes place is said to be in a refractory period, meaning a period of unresponsiveness. After the action potential occurs, the axonal membrane where it takes place is said to be in a refractory period, meaning a period of unresponsiveness. There are 2 types. The absolute refractory is a brief period of complete insensitivity to any stimuli that may cause an action potential to take place. No amount of stimulation can induce another action potential because the voltage-gated sodium channels are unresponsive. Following the absolute refractory period, the relative refractory period occurs. Only strong stimulation can produce another action potential because the flow of potassium ions out has temporarily hyperpolarized the neuron, so a stronger stimulus would be needed to reach a threshold. These action potentials don't just occur once as the message travels down the axon. It happens several times. The process we discussed in the previous slide is regenerated along the axon, meaning sodium channels open in sections down the axon until the electricity reaches the axon terminal. This has to occur because electricity would dissipate between the axon hillock and the axon terminals. Some of the electricity is lost, but if it is regenerated, the exact same message that originated from the axon hillock will be the same when it reaches the axon terminals. Action potentials can only occur down the axon, never the other direction, because the sodium channels enter that state of refractory as discussed in the last slide. The places in which the action potential is regenerated is at the nodes of Ranvier. Remember these? Nodes of Ranvier are the spaces in between myelinated sections of the axon. This is where the membrane of the axon is exposed and sodium, potassium, and NAK pump channels are located. So the action potential occurs at the node of RON-VA, goes down the axon to the next node, and is regenerated here again. Jumps to the next node, is regenerated again and again and again until it reaches the axon terminals. So again, action potentials occur at every node of RON-VA in order for the original message to stay the same until it reaches the axon terminals. The way the action potential travels down the axon is called saltatory conduction. You can see saltatory conduction taking place in this figure. You see the node in the first section and the action potential taking place. Sodium is rushing in, potassium rushing out, etc. The electricity passes through the myelinated section of the axon until it reaches the next node. You can also see by the red coloring that the action potential is losing its strength, thus it needs to be recharged. So, another action potential occurs at the next node. The action potential travels down the myelinated section of the axon to the next node, and the action potential occurs again. This is saltatory conduction. Now let's start looking at synapses and what happens when the action potential reaches the axon terminal. When the action potential reaches the axon terminal, it is converted into a chemical signal, neurotransmitter. As a reminder, neurotransmitter is the chemicals inside the synaptic vesicles in the axon terminals and serves as the basis of communication between two neurons, which will become apparent in a few moments. When the neurotransmitter is released into the synapse and bind to the receptors on the postsynaptic membrane, it alters the membrane potential of the postsynaptic cell. These changes that occur in the postsynaptic cell are called postsynaptic potentials, or PSP. No, I don't mean those portable PlayStations. Does anyone even play those things anymore? Neurotransmitters released into synapses briefly alter the membrane potential of the postsynaptic cell. These are PSPs, a given neuron receiving synapses from hundreds of other cells is subject to hundreds or thousands of PSPs. When integrated, this massive array of local potentials determines whether the neuron will reach threshold and therefore generate an action potential of its own. There are two types of PSPs: excitatory postsynaptic potentials, or EPSPs, and inhibitory postsynaptic potentials, or IPSPs. EPSPs contribute to the cell's probability of firing its own action potential down its axon. It depolarizes the cell, meaning it moves the neuron's polarity toward zero and thus toward the threshold of the neuron. IPSPs are the opposite. They cause the cell to hyperpolarize and decrease the polarity. It moves the membrane potential away from zero and thus away from the threshold. It contributes to the canceling of an action potential from taking place. Here we see three neurons. The yellow is the postsynaptic neuron, while the red and blue are the presynaptic neurons. The red neuron is sending EPSPs to the postsynaptic neuron. If there are enough EPSPs that accumulate in the neuron, it will reach its threshold and fire an action potential. The blue neuron is sending IPSPs to the postsynaptic neuron. It is decreasing the chance or canceling out the action potential from taking place because it is making the neuron more negative. Here is a more visual representation of how EPSPs and IPSPs influence a neuron's action potential. In figure A, only the red presynaptic neurons are active, and they give the postsynaptic neuron EPSPs, and thus the postsynaptic neuron's polarity increases and it fires its own action potentials and becoming the presynaptic neuron in the process. Figure B shows two neurons giving the postsynaptic neuron EPSPs and two neurons giving the postsynaptic neuron IPSPs, and thus canceling out the probability of an action potential. The postsynaptic neuron thus never fires an action potential. Figure C shows three neurons giving the postsynaptic neuron EPSPs and two neurons giving the postsynaptic neuron IPSPs. The EPSPs are overpowering the IPSPs because there are more of them, and thus the postsynaptic neuron reaches its threshold and fires an action potential. So it is usually the convergence of excitatory messages from many presynaptic neurons that is required for a neuron to fire an action potential. This summation of potentials from different physical locations across the cell body is called spatial summation. Postsynaptic effects that are not absolutely simultaneous can be summed because the postsynaptic potentials last a few milliseconds before fading away. The closer they are in time, the greater is the overlap and the more complete is the summation, which in this case is called summation. The EPSPs come from one source. If there is too much time in between the arrival of EPSPs, they will fade away and the neuron will never reach its threshold. Hey, I'm over here again for this slide. Okay, now let's take a look as to what happens when the action potential reaches the axon terminal and how it is converted into neurotransmitter. Stay with me on this one, okay? Now, the action potential reaches the axon terminal can change the polarity of the membrane. There are calcium ion channels on the axon terminal, and when the membrane polarity changes due to the action potential, they open up, letting in a flood of calcium ions into the axon terminal. The calcium forces exocytosis, which is the fusion of the synaptic vesicles with the presynaptic membrane. You can see this occurring on the figure. Follow the top left vesicle to the next. This calcium is forcing the synaptic vesicle to float toward the presynaptic membrane, and when it reaches it, it fuses with the membrane and opens up. Remember, there is neurotransmitter inside these synaptic vesicles, and thus, when they fuse with the presynaptic membrane and open up, they release the neurotransmitter into the synaptic cleft, the space between the presynaptic and postsynaptic membranes. The neurotransmitter then binds with the receptor proteins on the postsynaptic membrane. The receptors open up and let in an array of charged ions, creating either EPSPs or IPSPs. If the ions are positively charged, they create EPSPs. If the ions are negatively charged, they create IPSPs. This is how the action potential is converted into neurotransmitter, and this process is called synaptic transmission. But that's not all. Now we have a lot of leftover neurotransmitter in the synaptic cleft. What happens to it? Well, two things can occur. There are specialized enzymes in the extracellular fluid that are responsible for breaking down excess neurotransmitter. This process is called enzymatic degradation. But for the neurotransmitter that is still nearby or in the synaptic cleft, reuptake occurs. Neurotransmitter bind to specialized proteins called transporters on the presynaptic membrane and are brought back into the cell in a synaptic vesicle for recycling. The neurotransmitter will be used again at a later time. Also, along the axon terminal membrane are another type of protein called autoreceptors. When excess neurotransmitter binds with autoreceptors, this decreases the release of neurotransmitter into the synaptic cleft. Okay, you still with me? I know it sounds like a complicated process, just like the action potential. It may be hard to visualize it, so I have included a link in the description of the video that may help understanding synaptic transmission as well. When trying to understand how neurotransmitter binds to receptor proteins on the postsynaptic cell, think about a lock and key. Neurotransmitters can be considered the key. The binding sites as seen in the picture can be thought as the lock. The protein is the door, and the ion is you, the person trying to pass through the door. The neurotransmitter, the key, enters the lock, the binding site. Once the neurotransmitter binds to the receptor opens and allows ions to pass through. As you can see here, the ion is sodium, and being positively charged causes EPSPs to occur in the postsynaptic cell. If it were a chloride ion, which is negatively charged, it would cause an IPSP. Again, when synaptic transmission stops, degradation or reuptake occurs. Degradation is when specialized enzymes break down the neurotransmitter into its simpler components and dissolve it into the extracellular fluid. Reuptake occurs if the neurotransmitter enters the transporters on the presynaptic membrane to go back inside the synaptic vesicle for recycling and to undergo synaptic transmission again. For the most part, we have been discussing synapses that occur in the axodendritic form, meaning the axon terminal synapses with a dendritic spine or the dendrite itself. However, that is not the only place in which synapses occur. You've actually seen these in the previous slides on several of the figures. Axosomatic synapses are when the axon terminal synapses on the soma instead of the dendrite, while axonal synapses are when the axon terminal synapse on another neuron's axon. Everything is the same though. Action potentials and synaptic transmission still takes place. It's just the way the synapse is formed. Here they are more clear. The first figure shows an axodendritic synapse, the traditional and most common synapse. Figure B shows the axosomatic synapse, where the axon terminal synapses with the soma directly. Figure C shows axoaxonic, which is where the axon terminal synapses directly onto the axon of another neuron to send a combined message to the postsynaptic neuron. And figure D shows a dendrodendritic synapse where communication occurs over two dendrites. We won't be discussing these in this class, but I will eventually add a video over them in the future. Well, this wraps up the chapter 3 lecture. I'll see y'all online. Ciao. The black neurons rely on electrostatic pressure to bring potassium into the cell as well. They're an equilibrium or the resting what? You see, as three sodium what?